Today I will be discussing a hypothetical developmental network in which the pathway will lead to the formation of spikes in certain cells of the ectoderm for a spiny beetle. Developmental networks can be described as a pathway of gene products and how they interact with one another to cause traits in an organism. An arrow in the pathway represents activation and means the pathway progresses, while the T-bar represents inhibition or repression. Here is our pathway that we are given. As you can see, there are 5 different genes, S, P, I, K, E, that will influence whether there will be formation of spikes or not. The S gene will activate the P gene, P then activates the I gene, I will then repress the K gene, which is essential to the formation of spikes, as if K was not repressed, then the K gene would also repress the E gene, which would not lead to spikes. Because I represses K, the E gene is able to activate the formation of spikes. Now we will cover how certain mutant strains affect our pathway by looking at a few examples. For the first scenario, we have a mutated P gene. Since the P gene is not able to function properly, the I gene will not be activated and therefore cannot repress K. This results in K repressing E and will lead to no spikes in the ectoderm, but rather a smooth appearance. In the next case, we have a mutated I gene. For this pathway, I cannot function properly, so the K gene is again not inhibited or repressed, and just like before will result in no spikes, as K will ultimately inhibit the E gene again. What about if 2 genes are mutated? We call this a double mutant, and we'll look into these cases as well. For the first double mutant, we have mutations in both the I gene and E gene. For a shortcut, we know that this will automatically result in no spikes, as the E gene is essential in that formation. So this double mutation also leads to a smooth beetle. Lastly, we have double mutants of S and K genes. Since S is not able to function, P will not be activated, which leads to I not being activated either. I cannot inhibit K, but that does not matter much as we have a non-functioning K gene as well, which cannot inhibit E. We finally have a mutation that still results in spikes, as the E gene was not affected. Lastly, for a tidbit of extra knowledge, let's think about why not all the cells in the ectoderm have spikes. This could be because too many spikes will get into the way and affect their movement of the beetles, as well as only some parts of the ectoderm need spikes for protection purposes. Parts that would not need any spikes would be the underside of the beetle. Spikes will be present in the ectoderm of the beetle wherever it is needed.